Hello students, in the last video we have seen the immune response. So we have started immunity and we have seen the types of immunity, innate and acquired, and after that we have also seen the other type of immunity. We have seen the active and the passive immunity as well. And whenever we saw the immune responses, we understand how actually the primary and secondary responses work, and according to that B and T lymphocyte activates, right? So all that part is completed. So if you have not seen, I tell you once again, you just see that video, then only you will understand this part. Okay, so first of all we should know actually what is antigen and antibody. So before coming to all those things that are the responses, how antibody work, how antigen antibody reaction occurs, how we can able to kill the pathogen, everything we are going to study today. See what happened to the antigens and what are actually those antibody, what are their chemical nature, right? So first of all, if it comes to antigen, we write antigen A as Ag, and if it is coming to antibody, that we will write A as AB. Okay, so in small, just to reduce the time, we will use the word antigen as Ag and antibody as AB. Okay, so first of all, what are those antigens, right? So antigens are any type of foreign particle which can capable of inducing our immune system to produce the antibody. So due to the entry of all those foreign particle, if our immune system produces antibody against that foreign particle, then we can call that foreign particle that has invaded, that is known as antigen. Okay, so the antigens can be of anything. It can be a carbohydrate, that means it can be a polysaccharide. Okay, it can be a protein, it can be a conjugated protein also, because we know the protein, it can be completely protein, or with that protein part some non-proteinaceous compound can also attach, that is known as conjugated protein, like glycoprotein, lipoprotein, all those proteins are conjugated protein. So our antigen chemically, that means if you see the composition, then it can be a polysaccharide, that means it can be a carbohydrate, it can be a protein, or it can be a nucleic acid. Okay, so that is known as a antigen. So antigens actually is the foreign particle, but that, suppose this is a microorganism. Okay, so on that surface of the microorganism we will have some region that will be known as antigenic sites. Okay, so these are some sites with which the antibody can attach. Okay, so suppose this is completely, this is a foreign particle, this is a microorganism. Just think this organism is a bacteria. With that bacteria we are going to have some that means as the bacteria is a foreign particle, it will have some other properties. So on the surface of the bacteria, it is going to have those red colored structures which is going to attach with the antibody. Okay, so the antibody structure is a Y-shaped structure, right? So with that region the antibody attaches, that is known as the antigens. Okay, so you just need to know that antigens, maybe this antigen is not like it is a complete foreign particle. So what happened, this is just a specific region or attachment site of that foreign particle with which the antibody attaches. So this whole black colored substance, this will be a microorganism, right? This red colored structure, those patches, they are actually the antigenic site. Okay, these are the antigenic sites, and those blue colored Y-shaped structures, these are the antibodies. So these antigenic sites are those sites with which the antibody will attach. Okay, and it will carry out the antigen antibody reaction. Okay. So now antigens, the type, how many different types of antigens we can have? See, if it comes to the antigens, depending upon how actually actively it can produce the antibody, it is of two types. Okay, it is of two types. First type, it can be a complete antigen or it can be a incomplete antigen. Okay, so what are those complete antigens? Complete antigens are those antigens whenever it enters inside our body, our immune system produces the antibodies. So these antigens, the complete antigens, are those foreign particles which are able to or which are capable of inducing the antibody production from the B lymphocyte or the plasma cell. So whenever these antigens enter, our immune system produces the antibody that is known as the complete antigens. And what are those incomplete antigens? Incomplete antigens are those antigens which are the structures whenever it suppose directly enters inside our body, but it cannot induce our immune system to produce the antibody directly. Okay, suppose this is the incomplete antigen, right? Directly it cannot So those incomplete antibodies, whenever it enters inside our body, it cannot induce our immune system to produce the antibody. Rather, what it is going to do, it attach with some other particles. Okay, other suppose carrier particle, and whenever with the carrier particle this incomplete antigen attaches, then only it is capable to induce our immune system to produce the antibody. So these are the two different types of antigens we have seen, and also we can see other category where it is present inside our body or not. Okay, so those antigens which are present inside our body, which are not a foreign particle which is of our own, they are known as the self antigen, right? And those antigens or those foreign particle which is coming from outside, which is not of our own or of our own product, they are known as the non-self antigen. So you don't think that always the antigens will be foreign. Why? Because in our blood grouping system, in the body fluid and circulation of first year, you already know that the surface of the RBC have two type of antigens, antigen A and antigen B. So those antigens, they are the self antigen which is present on the surface of RBC that cause coagulation reaction or clotting whenever a different blood group enters or transfused, right? So those antigens which are present on our RBC surface, that are antigen A and antigen B on the surface of RBC, they are known as the self antigen. And what are the non-self antigens? Those foreign particles who come from outside, right? They are known as the non-self. They are known as the non-self antigens. Okay. Now after that, let us see the other type of compound which is produced by our body as a response of the foreign particle, that are known as the antibodies. So if you see composition wise, the antibodies, they are made up of amino acid. Okay. So these are the amino acid compound. So it is actually a structure which is going to have amino acids. So those amino acids molecule, they are attached to each other by a chain, that is peptide chain, and it forms the peptide structure. Now those antibodies, these antibodies are just some immunoglobulins. Okay, these are some of the immunoglobulins which are produced inside our body as a response of the invasion of a antigen. Okay, so that's why we can say that yes, all antibodies, they are the immunoglobulins, but all immunoglobulins are not the antigens. Now just see those antibodies. What is the different type of antibodies we can have? See, these antibodies will be of 5 different types. I will tell you what are those. We will see the 5 type of antibodies. What are they? First one, that is IgD, IgA, IgG, IgM and IgE. Okay, so 5 type of antibodies are present. And to understand the structure of the antibody, we will take a structure of one antibody, that is the IgG. Okay, so from all those 5 type of immunoglobulin G will be taken to study the structure of antibody. So I told already that those antibodies, they're made up of what? They're made up of amino acid. So let's just see the structure. This is one amino acid chain. So it's a peptide chain. Here there is another peptide chain. So this is the first peptide chain, right? So this is the first chain where the amino acids, they're attached with each other by a peptide bond, right? So this is one chain. In this structure of antibody, there will be another chain. That chain of that amino acids, they are smaller than this chain. So there are 4 chains, 4 polypeptide chains will be found. 2 of the polypeptide chains, they are longer, and 2 of the polypeptide chains, they are smaller. So, or smaller or shorter. So this is the another polypeptide chain. So this is the another polypeptide chain. So this will attach to the longer chain by disulfide bond, right? So in between this, the longer chain and the shorter chain, they will have the disulfide attraction. They will have disulfide bond, and because of that we will get a antibody structure. So this way they will get attached to the other. Okay, so this way we will get the structure of the antibody. So as that antibody will have 4 chains, 2 of them, this red color chains, they are the longer chain. Also we can call this chain as the heavy chain. Okay, so this is the heavy chain, and the shorter chains, they are also known as the light chain. Okay, and the heavy chain and the light chain, they are attached to each other by disulfide bond. Okay, so this is the disulfide bond. This is because of what? The light chain and the heavy chain can attach to each other and can form an antibody. So that's why the antibody structure is also known as the H2L2 structure. So in question it may be asked that antibodies are also known as H2L2. Give reason why. So this is the reason that this antibody is made up of 4 chains. 2 of the chains, they are the longer chain, they are also known as heavy chain, and 2 of the chains, they are shorter chain. So these are also known as the light chain. So as 2 of them, they are heavy, 2 of them, they are light. So that's why it is known as H2L2 site. Now to see in this antibody will have 2 regions also. If you see region wise, then 2 regions will be present. So what are those 2 regions? This is the first region, this is the another region. Okay, so this is the only region from where, from the region it can attach to an antigen, right? So this is actually known as the antigen binding region. Okay, and that's why this is also known as variable region. Okay, and this region where only the heavy chains are found, they are known as the constant region. Okay, they are known as the constant region, right? And here this region where we can see the antigens attaches to the antibody, what we can call, this is also known as FAB region. And this region, this is the constant region, also called as the FC region. So this region, this is actually fragment where the antigens attach to the antibody. So this antigen attachment region is known as FAB region, and where the antigens cannot attach, that will be known as the FC region or fragment crystallizable region. Now just see this antibody. Is it present normally like this structure, or some another way we can able to see? So just see this antibodies, these antibodies actually are produced by whom? This is produced by one cell, that is B lymphocyte. So this B lymphocyte, which I told already that B lymphocyte, they will form two cell, they will synthesize two cell. One cell, this is known as the plasma cell, and another is the memory cell. So that plasma cell will produce the antibody. So maybe it is possible that this plasma cell, whenever it produces these antibodies, they will directly go to the blood vessel or maybe to the lymph, and they can be present only like this. But very often this is also possible that on the surface of the B lymphocyte it can attach. Okay, so in the bloodstream, so this is suppose the bloodstream. So in the bloodstream directly this antibodies can be found freely flowing inside the blood, or maybe it can also be possible that it can attach, or it is present on the surface of the plasma cell or the B lymphocyte. It will always attach to the surface of the B lymphocyte. So one more thing I would like to tell you, that if you see the structure of the amino acid, what is the shape or what is the structure of the peptide chain? As you can remember that this amino acid will have a central carbon. It will have one side a carboxylic acid site and carboxylic acid group. Another side it will going to have a amino group. One side it is going to have hydrogen, and another region it is going to have a variable group. Now one amino acid can attach to the next amino acid by a peptide bond. So how this will be possible? That see, if I draw another amino acid, suppose the another amino acid is present here. So what will happen? One carboxylic acid group, one amino acid group, amino group, one region it is hydrogen, and another region is the variable group, right? So how the bond formation will take place? So bond formation will take place by this amino group and carboxylic acid group. So one will donate the electron, another will accepting it. So what will happen here? The region, this OH and one of the hydrogen, okay, that will bind and it will produce water molecule. And whenever this water molecule is removed, then what happen? A bond formation will take place, and that bond and this bond will be known as the peptide bond. It is not necessary, it is not needed. The intention is to make you understand that in this peptide chain, one side will be the C region where carboxylic acid is present, and another end of the same amino acid chain or peptide chain will have the amino group. So just see, suppose this is a peptide chain. So in that peptide chain, you will going to have one C region where carboxylic acid is present. So another region will be the amino acid region, so it will be a N-terminal. So always remember, if it is in case of a peptide, then this peptide chain will have a N-terminal and another will be the C-terminal. So this way one amino acid will attach to the next amino acid by the removal of the water molecule, and this way peptide chain will go on forming. And if you see the structure, where is the C terminal and where is the N terminal, right? So you can see that in this heavy and light chain, the FC region, fragment crystallizable region, this region is going to have the that C terminal, and exactly on opposite direction in this end it will going to have N region, right? Similarly, in the light chain also same thing is happening, that in the light chain we will have the C terminal on this region and N terminal on this region. And because of that, what happened, this amino group will try to make bond with some another structure. So that's why it is a variable region with which a foreign particle, that is an antigen, will try to always bind, right? So this way we will get the structure of the antibody. Hope we have understood. I hope I have told you what actually the structure have been formed. Now you can ask me where actually, from where does sulfide bond form or take place. See, one of the amino acid is the cysteine or methionine. They are sulfur containing amino acid. So if that sulfur containing amino acid, this variable group will have sulfur. Suppose here there is a sulfur, from here there is a sulfur. So in between the two sulfur bond formation will take place, and that's why this H2 and L2 structure can be bonded together and form a antibody. Okay, remember it's a protein. After that, let's just see the different type of immunoglobulins. Now after that, see the different types of antibodies. So as I already have told you that there are 5 type of antibodies are present. What are they? This is immunoglobulin A, immunoglobulin D, immunoglobulin E, G and M. So all of them, they are going to have a FAB region, FAB region. They are going to be different. Okay. So if you see all of the immunoglobulins, then the shape will be different. First of all, if you see the immunoglobulin A, then this immunoglobulin A will have 2 Y-shaped structure, and whenever we see this 2 Y-shaped structure, so this 2 Y-shaped structure will attach to each other by another chain that is known as J chain. In case of immunoglobulin A, M also, it is also going to have the another J chain. So J chain will help attaching the 2 Y-shaped structure of immunoglobulin A and immunoglobulin M. The next structure, just see how many FAB region is present. So if you see only one Y-shaped structure, one antibody, then in one antibody there will be 2 FAB region. So if you see this immunoglobulin A, then how many FAB region will be present? 1, 2, 3, 4. Right? If you see immunoglobulin D, then it is going to have 2 FAB. In case of immunoglobulin E, also it is 2 FAB, and immunoglobulin G, it is also 2 FAB region will be present, with which it is going to attach to the antigens. If you see immunoglobulin M, there will be 10. 4, 5, 6, 7, 8, 9, 10. So 10 FAB region, FAB region will be present, with which one immunoglobulin, that immunoglobulin M, can attach to the antigens. So if you see the structure wise, then which is the smallest antibody or immunoglobulin? That is immunoglobulin G. Remember, immunoglobulin G is the smallest antibody, and immunoglobulin M, as it is given in the diagram, it is the largest antibody or immunoglobulin. Now just see the percentage. So this question came a number of times, that what is the largest or maximum number of antibodies are. So which antibody is present in maximum in our body? That is the IgG, which is the smallest, that is present in highest amount also. Now the next one, which is present in very less amount, that is IgE, which is even less than 0.1%. Now just see the rest of them. IgA is around 10 to 15%. If it is IgD, it is around 0 to 0.2%. E already it is less than 1%. Highest amount, that is 80%, that is IgG, and IgM is around 5 to 10%. Now the next one, if you see location wise, then where all those immunoglobulins can be found. If you see immunoglobulin A, it is found in the tear, saliva and colostrum. Remember, already in the human reproduction chapter, we already got it that in the mother's milk, we will get the immunoglobulin A. Next one, see, if you see IgD, this is present on the B cell surface. I told already that this immunoglobulin not present as an individual structure. It can be also attached to the B lymphocyte. So not only this IgD, but there is another one, that is IgM, which can also attach to the B cell surface. Now, next one, this IgD that can be also found in the blood and the lymph. And if you see IgE, that is found in the blood, and also it can be bound to the mast cell. Mast cell, remember, mast cell is actually one type, this is basophil itself, which release three chemicals. They are heparin, histamine and serotonin. Now after that, this immunoglobulin G, this is found in the blood, lymph as well as in the intestinal region, that is immunoglobulin G is present. And if you see IgM, it is found in the blood, lymph and B cell surface, it is present. Now the most important thing is their function. So from here, these 2 points are going to be very much important. This is 80%, this is 0.1%. This is going to be very much important. Now after that, this is going to be important in colostrum, which antibody is present, not from only this chapter, also from human reproduction chapter. Next one, if you see function wise, then what is the function of immunoglobulin A? It will help in only external function, like it is in the saliva, in the tear it is present. So if some foreign particles suppose try to enter inside your eye, then this IgA will kill that pathogen. Now if it is IgD, it will help in antigen recognition by beta cell, B lymphocyte. So it will help basically in recognition. Okay. Next one, recognition means one thing you should know. Suppose in the blood vessel, this is the blood vessel. Now a foreign particle is entering. So whenever that foreign particle will enter, then this IgD will attach. Okay, and it will represent or recognize itself as or it will just tell or induce the immune system so that other specific cell can come to that region and can kill the pathogen. So basically it helps in opsonization. So what is opsonization? I will tell you later. So we'll see after that IgE, which will help in allergic reaction. See, there is hypersensitivity. When an individual have allergic reaction, then suddenly the individual will have high level of IgE. So IgE is actually one indicator of hypersensitivity. If you go to a doctor to see whether you have allergic reaction or what may be the reason for your body imbalance or some reaction or irritation is happening, then in the blood test it is detected IgE that is increasing in our blood. So in allergic reaction we will have high level of IgE. Next one, if you see IgG, it will help in complement activation. Remember, in the innate immunity we have complement system which is having some proteins which get activated by cascade mechanism. So complement activation, complement system will be activated by IgG as well as IgM. Okay. So that is all about the different type of antibodies and their function. So all of these things are going to be very much important. You have to write and remember also. Let me tell you one important thing, that from all the type of immunoglobulin, which is the smallest one, that is IgG. And that's why through the placenta from the mother's blood to the fetal blood, only IgG, that is only the transplacental immunoglobulin, can enter into the fetus body, which provide immunity to the baby, to the fetus. In the embryonic state even it will enter. So after that, let us see how this antibody will function. So all this mechanism we'll see after that. Now after that, just see the last part of this video, that is antigen antibody reaction. So if an antibody invade, or suppose an antigen have invaded, then the antibody have to kill that foreign particle, or it actually should develop the immune response. So how it will develop the immune response? That will be by 5 steps or 5 different types. It can cause the lysis, or it can kill the pathogen, or if it cannot kill the pathogen, then at least it can recognize, or it can help the immune system to recognize the pathogen. Okay, so let us see the 5 different types of mechanism by which the antibody react with the antigen, and that 5 phenomenon, they are actually grouped under the types of antigen antibody reaction. So under that antigen antibody reaction, the first type of antigen antibody reaction is agglutination. So whenever the foreign particle have entered, suppose this is the blood vessel, okay, and in that blood vessel, suppose in that blood vessel a number of foreign particles, they have invaded, okay, suppose these are the foreign particle. So those foreign particles, whenever it enters, it can freely spread and can go to the tissues. So that's why before they can spread into our body parts or the tissues, and it can go to the other regions or infect our other body parts, before that, what the antibodies are going to do, those antibodies will clot all those pathogens. Okay, so like this, suppose these are the antibodies. So antibodies will get clotted by this agglutination reaction. So this is one phenomenon by which the reaction will take place. The antigen antibody reaction will take place, where suppose those pathogens are present in our blood vessel, then before it can spread into our tissues, this antibodies will clot, or it will help in clumping, or it will help in coagulation, or it will help in agglutination. So this is known as agglutination reaction. The next one, that is opsonization. Now what is opsonization? Opsonization means labeling. Okay, so what is labeling? See, agglutination means it is clumping. Okay, remember opsonizations mean this is labeling. Now labeling means what? See, some of the antibody may not kill the pathogen or the antigens, but at least it can do one thing, that it can tell the immune system that there is a foreign particle, and send the immune system some toxic cell to kill that pathogen. So there is a foreign particle freely flowing. The other cells of the immune system is not able to recognize that pathogen. Then what will happen? There will be an antibody who can understand that yes, this is a pathogen. So what it is going to do? It will going to have the reaction opsonization. So by this opsonization reaction, what it is going to do? It is going to attach to it. Okay, and whenever it is attaching to it, it will stimulate the immune system to produce the toxic cell or cytotoxic killer cell. So it will induce the immune system to produce a cytotoxic killer cell to come to that region and kill that foreign particle or the antigen. So that reaction is known as opsonization reaction. After that, see, the next reaction is precipitation reaction. See, in this precipitation reaction, what happened? This is the blood vessel. Now, there may be some type of pathogens which are very much small in their molecular weight. So, suppose these are the pathogens and these are the antigens. And these antigens have very less molecular weight, because of what? It can directly lyse, or they can directly dissolve in the blood or plasma or lymph. So if it can dissolve in the blood, then it is not possible that the macrophage or the cytotoxic killer cell can break and can kill that foreign particle or antigen. So before it gets dissolved in the blood or the lymph, what our antibodies will do? These antibodies will attach to it. So whenever those antibodies attach to it, attach to those antigens, then what will happen? Molecular weight will increase. So it will help increasing the molecular weight. So molecular weight will be increased. The antigen can no more dissolve in the blood, and that's after that the antibodies or other cytotoxic killer cell can come to that region and can kill that foreign particle or the antigens. So that precipitation reaction is that reaction will occur for those antigens which are of very less molecular weight. So those which have very less molecular weight, it is not possible that the cytotoxic killer cell or the macrophages can attach and can kill those foreign particles. So before it can dissolve in our blood or lymph, before it dissolves, what our antibodies will do, it can attach to the antibodies who are just about to get dissolved. So by this, they will increase the molecular weight. The antigens can no more dissolve in the blood or lymph, and this way the immune system can work on that pathogen. So that is known as precipitation reaction, which will increase the molecular weight and preventing those foreign particles from dissolving into the blood or lymph. After that, the next one, see, this is the neutralization. So what is the neutralization reaction? Suppose this is the bloodstream, right? Here we have the tissues. Now what happens? Some foreign particle. I have told you that antigens may not be the whole foreign particle. So it may be a microorganism where some antigenic site is present who can recognize itself as a foreign particle. So what happened? Those foreign particle will have some virulent site. Okay, suppose this is a foreign particle microorganism, and in that microorganism's body some antigenic sites or some virulent sites are present. Those virulent sites can cause reaction in our body and can kill our cell, and this can be pathogenic to our body. Okay, and if you kill or if you can destroy those pathogenic sites, then maybe that microorganism will lose the capacity to infect us. Okay, so this is the infectious region with which a microorganism can infect our body cells or the tissues. So some of the antibodies can come to that region. It may not kill that pathogen, it may not kill the microorganism, but at least it can break that virulent site. And whenever it breaks the virulent region, then this microorganism become non-virulent. It can no more can infect our body cells or the tissues. So this is a way by which we can able to neutralize a virulent type of pathogen into a non-virulent type of pathogen. So this is the neutralization reaction, where actually it will inactivate the pathogen. After that come to the last part, that is lysis. Lysis means it is completely going to break the foreign particle. How it is going to break the foreign particle? So this is a antigen, right? So this antigen will come in connection with a macrophage, or suppose a type of antibody. So whenever that antibody come to that region, so that antibody can now completely make whole. Some type of antibodies are so strong that it can completely break or can lyse the foreign particle into some pieces. So whenever that antibodies work on that foreign particle or the antigens, completely that antigens will break. Okay, it will cause lysis, and once it causes lysis, the antigens will die. So there are different ways. We have a number of ways by which we can able to kill that pathogen. Lysis means it is going to break that foreign particle. The whole cells, it will be caused by some very strong antibodies. So by some strong antibodies, it can cause lysis of the foreign particle, and this way we can able to kill that antigen. So there are 5 different types by which we can able to have the antigenic or antibody reaction, and we can able to prevent ourselves from the foreign particle infection. So that is all about this antigen antibody reaction. Other parts you just see sequentially one after the another video so that you can understand, it make easier for you. So that is all about the video. Hope you have understood. Thank you.